Assalamu alaikum. I'm Dr. Sidra Arshad with the fifth lecture in the Physiology of Special Senses, and today I shall be discussing the physiology of two chemical senses that are taste and smell. We'll start by discussing the taste sensation, so let's just begin. Sense of taste is basically the function of taste buds in the mouth. Taste buds are the regions where the taste receptor cells are present. The sensation of taste, it is influenced by a lot of other factors, which include the smell of the food which is being taken, texture of the food, the temperature, certain psychological factors that where a person is at the moment when he or she is taking their food, the surroundings, and the presence of irritating substances in the food, which can alter the entire experience of the taste. So sense of taste, it is important for avoiding potentially dangerous substances. It is seen most of the dangerous substances, the poisons, they have a bitter taste. So these are usually avoided and ingesting pleasant and nutritious ones. So the diets which are nutritious, they are usually pleasant tasting. So they are preferred over the unpleasant tasting substances. So this entire phenomenon is known as taste preference. It is a complex process which involves the CNS as well. And through this process, an individual selects the best possible tasting food substances. So this is the importance of taste in a human being. So let's look at the primary taste sensations. So the primary taste sensations are sour, salty, sweet, bitter, and umami. So there is a concept which is known as taste threshold. So obviously, taste and molecules of different taste modalities are going to have different thresholds for detection or perception. As it can be seen, the bitter taste is going to have the lowest threshold. There is high sensitivity to bitter taste, and that is why the bitter taste is used to check for taste blindness. Certain individuals who may be blind to the taste, bitter compounds are usually used. In that way, it can be assessed whether they are completely insensitive to taste sensation or not. For this purposes, a special compound which is known as phenyl thiocarbamide is usually used. So this compound is used to assess for taste blindness. Another concept is taste index. So basically, taste index is the inverse of taste threshold. Different taste indices are taken with one primary molecule in each category of the taste, then the indices are compared with that molecule. This is the structure of the taste bud. Associated with the taste buds are the taste cells, which are the receptor cells for the taste sensation. You can see right over here in this diagram, these are the taste cells in the dark violet color. So the taste cells, they are present in the taste bud, interspersed between the supporting or sustentacular cells. The taste cells are specialized cells because on their apices are present microvilli. These microvilli, they contain the receptors for the taster molecules to bind, and when the tasters, they bind with the receptors present on the taste hairs or these microvilli, the taste cell is depolarized, and this depolarization is going to cause the release of neurotransmitter, which is then going to generate action potentials in the sensory afferent nerve fibers. So these taste cells, these are richly innervated by the sensory afferent nerve fibers, which are the cranial nerve, which carry taste sensations. So these microvilli, they are projecting onto the surface of the tongue in taste pores. So these are projecting in the taste pores, and this is increasing the surface area for the taste ends to bind with the receptors. Taste buds are found in association with the papillae of the tongue. The foliate papillae, which are found on the lateral aspects of the tongue, they contain a moderate number of taste buds. A large amount of taste buds are present in the circumvallate papillae, which are forming this V structure on the posterior surface of the tongue, and on the interior surface of the tongue are located fungiform papillae, which contain moderate number of taste buds. So all over the tongue, all types of taste ends can be perceived. However, they may be aggregated at certain places. The taste receptors are specialized receptors, and they are specific for each taste modality. So let's look at the taste modality and few examples of the common substances which are perceived as that specific taste. And then we are going to look at the receptor types for that taste modality and the mechanisms of signal transduction in the which is going to occur when these receptors are activated. So for salty taste, a few examples are sodium fluoride, calcium fluoride, and potassium fluoride. The receptor types are epithelial sodium channels, amongst others. The transduction mechanisms include these channels are ligand gated and they cause the influx of sodium ions, bringing about depolarization and release of the neurotransmitter. For the sour taste, the substances they are HCl, tartaric acid, citric acid, and carbonic acid. So these basically are the acidic substances. The receptor types for these tastes are epithelial sodium channels once again, and then there are hydrogen channels which cause the influx of hydrogen ions. There are specialized potassium sensitive channels which close when the sour tasted molecules they bind with the receptors, and by closing they are going to cause the depolarization in the taste cells. So the transduction mechanisms are they are ligand gated, cause either the hydrogen influx or they cause the blockage of potassium sensitive channels. Tasting substances they include sucrose and saccharin, for example. The receptor types are taste 1 receptor 2 types and taste 1 receptor 3 subtype. So these are G protein linked receptors, and they are the receptor types are specialized taste receptors 2 and 3. For umami taste, the substances are glutamate and aspartate. These receptors are also G protein linked receptors, and they include taste 1 receptor 1 and taste 1 receptor 3. So these are the subtypes which are involved in the binding of the umami tasters. The transduction mechanisms they include G protein coupled receptors. These are, and they are also the metabotropic glutamate receptor subtype 4. For the bitter taste, example is good enough example is quinine. The receptor types involved are T2 receptors. So T2 receptor is involved with the bitter taste, and this receptor has 30 different subtypes. So 30 different subtypes of T2 receptors are found, and these receptors are once again G protein coupled receptors. This G protein is known as gusducin. Now that we have talked about the receptors, let's trace the taste pathway with how the taste sensation is all over to the central nervous system. So taste receptor cells of the taste bud, they detect tasted molecule as it binds to the receptors which are found in the microvilli of the taste cells. So when the taste molecules, they bind with their receptors, the signal transduction and subsequent depolarization is going to cause the release of neurotransmitter. This release of neurotransmitter is then going to cause the action potentials in the relevant nerves. So from the anterior 2/3 of the tongue, the taste sensations are carried in the cauda tympani branch of 7th cranial nerve. So from the anterior 2/3 of the tongue, the fibers in the lingual nerve, they enter the cauda tympani branch of the 7th cranial nerve, while from the posterior 1/3 of the tongue, mouth and throat, these nerve fibers, they are carried in the 9th cranial nerve. So from the posterior 1/3, 9th cranial nerve, which is the glossopharyngeal nerve, and from the anterior 2/3 of the tongue, it is the cauda tympani branch of the 7th cranial nerve. So these nerves, they then synapse in the nucleus of rectus solitarius. From the nucleus of rectus solitarius, the impulses, they pass to the ventral posteromedial nucleus of the thalamus. These fibers, they then relay in the ventral posteromedial nucleus of the thalamus. And from the ventral posteromedial nucleus of the thalamus, the third order neurons, they synapse in the gustatory area in the cortex. So the gustatory area, it is found in the frontal operculum, anterior insular cortex, and tip of postcentral gyrus. So these areas, they together form the gustatory area in the cortex. Some fibers from the nucleus of rectus solitarius, they also synapse in the superior and inferior salivatory nuclei, and through these mechanisms, the rate of production or secretion of saliva is also controlled when certain taste and molecules they bind with the receptors. For example, in cases of sour taste, more saliva is produced. So this is the taste pathway. So adaptation to taste sensation, it occurs in a matter of a few seconds, and this adaptation, it occurs at the level of the receptor and in the CNS as well. So this adaptation mechanism is different than that seen in the adaptation of certain cutaneous receptors, and this mechanism of adaptation is also applicable to the adaptation to olfaction. So now let's look at the abnormalities of taste sensation. These are a few abnormalities which may be found in certain conditions. These abnormalities include acusia, which is the complete loss of taste sensation, may be seen in cases of head trauma with viral infections, certain medications, and in zinc deficiency. Hypoacusia, which is the reduced ability to taste, it may be found with aging, smoking, radiation therapy, and certain medications. Dysgeusia, which is the distorted or unpleasant taste that the normally tasting substances, they are now tasting unpleasant, found in upper respiratory infections, oral infections with certain medications, and in those who are receiving chemotherapy. We have concluded our discussion on the taste sense. Let's talk about the sense of smell or olfaction. So unlike the taste sensation, the primary sensations of smell, they are not categorized as easily because there are so many classifications due to more than 100 primary smells which can be perceived by a human nose. Along with that, there are more than 1,000 receptors for different olfactory molecules. So therefore, the primary sensations of smell, they cannot be discerned. However, they are categorized or clumped together into certain categories, and these categories are usually used. For example, all these pleasant smelling or flowery smelling substances, molecules, they are categorized into the floral smell. Let's look at the olfactory membrane. So the olfactory membrane is that specialized region of the nose which is involved in the detection of these odorant molecules. Olfactory membrane is found in the superior part of each nostril. It extends from the upper part of the nose up to the superior turbinate, where it folds over that turbinate and it almost reaches the middle turbinate as well. The total surface area of the olfactory membrane is 5 square centimeters. There are more than 100 million olfactory cells in the olfactory membrane. These olfactory cells are actually bipolar nerve cells. So this is the organization of the olfactory membrane. You can see right over here, these are the olfactory cells interspersed between the supporting and sustentacular cells. The olfactory cells, they extend olfactory cilia into the mucus layer of the nose. These olfactory cilia are the regions where the olfactory receptors are present and the odorant molecules, they bind to these olfactory cilia. You can see there is this Bowman's gland which secrete the mucus, which is going to concentrate the odorant molecules in this layer, which they can bind with the olfactory receptors in the olfactory cilia. So these are the bipolar cells. These are the olfactory cilia, which these odorant molecules can bind. This is the organization. So this is the organization of olfactory mucosa. You can see that these olfactory cells, they have these two processes. The dendrites of the olfactory cells, they have the cilia which are projecting into the mucus layer. On the cilia are present the olfactory receptors, and then these axons of olfactory cells, they are moving into the olfactory bulb through the cribriform plate of. So in order to be perceived as smell, the odorant molecules, they must be volatile, they must be both lipid and water soluble so that they can reach the olfactory membrane and cross the mucus layer to bind to their receptors in the olfactory cilia. Aiding them are other molecules which are found in the olfactory membrane. These molecules are known as olfactory binding proteins. They concentrate the odorant molecules in the olfactory membrane so that they can bind with their receptors. So olfactory receptors are G protein coupled receptors, G stimulatory proteins, so that when the odor molecule it binds with the odor receptor, there are increased cyclic AMP levels, which cause the influx of sodium ions and other positive ions, which are going to bring about depolarization of the olfactory cells. So let's trace that pathway. Olfactory receptors, they are stimulated axons of the olfactory cells. They synapse with mitral and tufted cells in the olfactory bulb. So there are specialized structures in the olfactory bulb, which are known as the olfactory glomeruli. Glomeruli are the region of synapse between the olfactory cells, the axons of the olfactory cells, and the mitral and tufted cells. They're dendrites of the mitral and tufted cells. So these ball-like structures, these are known as the olfactory glomeruli. From there, the axons of mitral and tufted cells, they pass through the olfactory tract and olfactory stria, from where they divide into two pathways to terminate either directly in the medial and old lateral olfactory areas or via dorsomedial thalamic nucleus to newer olfactory systems. So we are going to see what these older and newer olfactory systems they are. Now let's look at the organization inside the olfactory bulb. So the olfactory bulb, it contains the axons of bipolar olfactory cells along with the tufted and mitral cells. Then there are periglomerular cells and granule cells. So you can see right over here, this is the olfactory glomerulus. This olfactory glomerulus is composed of axons of bipolar olfactory cells along with the dendrites of mitral and tufted cells. So these are the olfactory glomeruli. The olfactory glomeruli, in between those are present periglomerular cells, which are inhibitory cells, and in this way they are going to increase the sharpness of the olfactory sensation which is going to be perceived. Also present are the granule cells, which are also inhibitory cells. These can inhibit the mitral and tufted cells. These cells are stimulated by the feedback impulses from the higher centers, and these granule cells then are going to inhibit the mitral and tufted cells. These are involved in the adaptation, central adaptation mechanism. Now let's look at the olfactory areas. The medial olfactory areas are concerned with the basic behaviors related to olfaction. These include septal nuclei, which feed to hypothalamus and other limbic primitive areas. The lateral olfactory areas, on the other hand, they are divided into two systems, the less old system and the newer system. In the less old system, these areas are included prepiriform cortex, piriform cortex, amygdaloid nuclei, and enteromedial portion of the temporal lobe. As far as the newer system is concerned, it involves the relaying in dorsomedial nucleus of the thalamus. For the rest of the areas, the thalamus as a relay station is not involved in the less old lateral olfactory system and in the medial olfactory system. However, in this newer system, as part of the lateral olfactory areas, the fibers, they relay in the dorsomedial nucleus of the thalamus, and from there the impulses are carried to the orbitofrontal cortex. So this is the organization of the lateral olfactory areas. Before looking at the functions of these areas, let's talk about the adaptation of olfactory sensations. So you must know from your personal experience that smell sensations or olfaction, it adapts to extinction almost within a few seconds or a minute. Olfactory receptors themselves, they adapt about 50% in the first second, that they rapidly adapt to 50% in the first second, and then they adapt slowly. However, additional adaptation, it occurs in the CNS. One of those mechanisms involves the granule cells, that when the smell sensations, they are being perceived, the feedback inhibitory signals, they pass via granule cells where they inhibit the mitral and tufted cells. This is the mechanism of adaptation of olfactory receptors, which involve the receptor itself along with the adaptation which occurs in the CNS. So these are the functions of the olfactory areas which we had just discussed. The medial olfactory area is associated with basic behaviors related to olfaction, such as licking, salivation, and feeding behaviors, which are seen in animals as well. So the lateral olfactory areas, these are actually involved in complicated olfactory condition reflexes. Orbitofrontal cortex is involved in the conscious analysis of odor. This is the newer pathway. This is involved in conscious analysis of odor, that is, the odor, it can be perceived and categorized into different categories. Integration of olfactory inputs with other sensory modalities, it occurs in this orbitofrontal cortex. Amygdala is involved in the emotional responses to olfactory stimulation. Entorhinal cortex is involved in olfactory memories, and hippocampus is involved in the olfactory learning experiences. So these are the functions of the olfactory areas. Certain abnormalities may be seen with olfaction. These include anosmia, hyposmia, parosmia, hyperosmia, and age-related decline in smell. So anosmia means complete loss of smell. It may be seen in sinusitis, nasal polyps, head trauma, viral infections, and with certain medications along with aging. Hyposmia is the reduced ability to smell. It may be seen in sinusitis, nasal congestion, allergies, head trauma, and with certain medications. Parosmia is the distorted sense of smell, which means that the normal odorant molecules, they are perceived differently. The parosmia, it may be seen with head trauma, viral infections, nasal damage, and certain medications. Hyperosmia is the increased sensitivity to smell, which occurs in pregnancy, with migraines, and with medications. So it is seen that the ability to perceive smell, it also depends upon the certain factors which include age and gender. Age-related decline in smell, it may be seen because of the gradual loss of smell with aging. There is decreased ability to perceive smells with aging. Potential causes include natural aging processes in which degenerations of olfactory receptors may be occurring, and other causes which lead to degeneration of olfactory receptors may. Age-related decline in smell may be seen because of degeneration of olfactory receptors. So these are some of the abnormalities of olfaction. These are the study resources which were used to prepare these lecture slides, and you can use them to study on your own. If you have any questions, please contact us on our social media handles. Thank you so much.